Thank you very much for the introduction. I'm very excited to be here today to share with you our work that's mainly focused on human blood-brain barrier organoids for the purpose of modeling therapeutic delivery into the brain. So I'll start by giving a little bit of introduction about the blood-brain barrier. I think most of these audience would already know or have come across the term. The blood-brain barrier is essentially the very highly specialized the blood-brain barrier is a very highly specialized vascular system of the brain that's really designed to protect the brain from any potential harmful substances that might have entered the body. Now, unfortunately, when it comes to therapeutic delivery, it's estimated about 98% of drugs cannot actually cross the blood-brain barrier and then therefore cannot enter the target site. So there's a great clinical need that needs to be overcome. That is, the therapies that are developed for treating brain diseases need to be able to cross the blood-brain barrier. Now, in vitro models has really come in very handy and played very important roles for us to help us understand the human biology of the blood-brain barrier, as well as to facilitate drug development in the field of pharmacology development. Now, there are several in vitro blood-brain barrier models that currently exist. Perhaps the most traditional and very widely used and widely known model is the Transwell model. Now, this is the 2D platform where essentially monocultures of endothelial cells are plated on the insert of the transwell, and other neurovascular unit cell types, brain cell types, are cultured also as a monoculture, as a monolayer on the basal side of the transwell. And how people usually use this is by looking at whether or not the drugs of interest can actually cross the monolayer of endothelial cells from the insert to the bottom layer. Now, this model, even though it's very widely accessible and convenient to use, there is a lot of very well-known limitations and disadvantages to it. Mainly, the biggest one probably is that it's highly inherently leaky. And since then, many newer in vitro models have been developed, mainly 3D models that have been becoming more increasingly popular, and these include microfluidic chips, the hollow fiber apparatus, and bioprinting strategies. Now, these models are really great for reproducing blood-brain barrier functions, but as you can see, their fabrication can be relatively complex, and a lot of them require some extensive instrumentation as well to make them or to use them. So you can see that these models, while they're really great, they're not very widely accessible to most laboratories. So what we're introducing here is the 3D human blood-brain barrier organoids as kind of maybe a happy medium that's very reproducible in terms of its ability to model the blood-brain barrier, how we form these human blood-brain barrier organoids, that we take human astrocytes, human brain parasites, and human brain endothelial cells, and we put them together, and then they come together, they self-assemble into these spheroids that are about 200 to 300 microns in size. You can see that they actually interact directly with one another. So there's direct cell-cell interaction between all these three cell types. They're very easy to culture because all you need are these cells, and then you put them together, and then they kind of form on their own. And they're very, very high throughput in that you can culture up to thousands at a time if you like. They're very cost effective. You don't require any complex instrumentation or fabrication. And they're macroscopic in size. You can see them with your eyes without a microscope. Here's in relation, there's a PCR tube, and you can see how the organoids look like in relation to the PCR tube. We validated and saw that it produces BBB biology and functions, and it's really ideal for using in early screening because you can use it in a high throughput fashion to reduce risk at later stages during in vivo analysis. So some of the BBB markers that we have validated in the organoids include tight junctions and efflux pumps. On the left here, you can see that these organoids really express a high level of tight junctions and efflux pump with the green signal, as well as they also have the tight junction efflux pump function. When you inhibit these, you can see that they stop functioning in the organoid, but then they are able to, sorry, I'll move on from there. In the middle part, we have validated that the BBB organoids actually express a much higher level of tight junctions and efflux pump compared to the traditional Transwell model. So we're very happy with the reproduction of the BBB markers in our organoid system. One thing that we did next was to demonstrate the high throughput capacity of these BBB organoids by screening a large array of cell-penetrating peptides. So here we wanted to test if these cell-penetrating peptides can actually cross the blood-brain barrier as well. So we tested 18 cell-penetrating peptides in our demonstration here, and this is a waterfall plot showing the data that we have ranked when we ranked with the organoids. The higher the data means that it can cross the blood-brain barrier better. So within this set of data, we focus on the top 5 and move forward for further analysis. What we did next was to incubate the organoids with the cell-penetrating peptide as well as dextran. And this functioned as a very nice negative control because the dextran, if it's not able to cross the blood-brain barrier, but the cell-penetrating peptide is, it means that the cell-penetrating peptide being present does not disrupt blood-brain barrier function. And over here, we see that in the presence of NGRIL2, out of all the cell-penetrating peptides, we saw that in the presence of NGRIL2, somehow there is also a very high level of dextran influx into the organoids. And this leads us to believe that perhaps NGRIL2 was disrupting the function of the blood-brain barrier, and so we excluded it from other analysis, from future analysis. So what we did then was to move into our mouse model to confirm that these cell-penetrating peptides can actually cross the blood-brain barrier, and indeed we did see that all 4 of them were able to enter the mouse brain. We then validated our platform to find that it's compatible with mass spectrometry imaging as well. So what we used here is a positive control drug, BKM120, that we know can cross the blood-brain barrier. And we also picked a negative control drug, dabrafenib, that we know cannot cross the blood-brain barrier. And you can see very well here that this green signal here indicates that we can really detect a high level of BKM120 into the organoids, but dabrafenib, really, we cannot detect anything at all as expected. What we went on to show next is that our model is also compatible with inductively coupled plasma mass spectrometry, or ICP-MS, in that we were able to use this strategy to detect an anti-cancer drug called platinum. So what we have done here is actually conjugated platinum onto a BBB penetrating peptide called M13, and we showed that we are able to improve its ability to penetrate the BBB in our organoid system, and we later showed it also that it was able to penetrate the blood-brain barrier in mice. Our next strategy here that I'm showing is that we were able to model BBB penetration, model gene delivery to show BBB penetration here. So here we use an AAV that we've modified that can cross the blood-brain barrier, and you can see very clearly here that the modified version of the AAV has a significant level of influx in the BBB organoid compared to the control. We were able to translate this data into mouse as well, where we injected the AAV into mice, and we saw a high level of transduction in the mouse brain, as you can see in the green signal here, transducing mouse brain tissue. And we were also able to see similar effect, the same results in non-human primate models, with the green cells signifying AAV transducing the monkey brain. So some of our next steps towards licensing and commercialization, we really want to use this BBB organoid platform as a sort of drug development pipeline. We want to start off with a wide range of therapeutics that we can screen in a very high throughput setting with the BBB organoids, and this would really help us narrow down what we should bring forward in our in vivo analysis, either in rodents or in primates, and hopefully all this will lead us towards closer to human clinical trials. What we'll also want to use this BBB organoids for is to be able to study the mechanisms that would also help facilitate our knowledge that would help in clinical trials. Now, we're currently in the process of developing BBB organoids under disease-specific conditions, including tumors. brain tumors. We are also wanting to offer this as an ultra-high throughput screening strategy to look at a variety of BBB permeable compounds. And ultimately, we want to produce a kit, perhaps, that can be widely accessible to the scientific community, that they can screen whatever compound that they might be interested in. So with that, I'll conclude my presentation, and thank you for your time.